So we shall start the workshop in order to avoid further delays. And I'm very happy to open this workshop. This is the first workshop on applied biosensing, organized by UDESC, this campus, Technological Center of the UDESC, and sponsored by FAPESC. It's our state agency here in the state of Santa Catarina. It's also supported by the University of Oslo, who paid the registration system, and also supported by Minipa. It's a company who are paying the coffee break for Monday, which is today. My name is Pedro. I'm the full professor of the State University of Santa Catarina over the last 20 years. I'm very honored to open this event, and I'm delighted to invite here with me to share this opening ceremony the director of the campus, Professor Dr. Antônio Ronaldo de Souza. Thank you for, and you may come here, please. Thank you. So, the development of new materials, diminutive variable measuring system, artificial intelligence advancements have propelled the widespread use of biosensors. So the main objective of the workshop on applied biosensing is to provide a unique environment for researchers to discuss and exchange experience across the biosensor chain. So this international free forum, by the way, covers application in various fields, including biomedical, computational engineering, medicine, environment, biology, chemistry, through lectures, tutorials, full papers, expanded abstract, and posters. Full papers are invited to submit to the Journal of Physics conference series, but also are invited to send to the International Journal of Electrical Bioimpedance, which is the editor of the journal is here, Professor Orion Martinsen from the University of Oslo. Full papers. We warmly welcome you also to the Women in Sensors, supported by IEEE, International Electronic Engineering from USA. This is supposed to be the 5th edition of the Women in Sensor that we call WISE. This platform celebrates the pivotal role of women in sensor technology and biosensing applications. So the event features 4 lectures. The event will be on the 5th of June, which is on Wednesday. 1 mini course, which we're going to deliver afternoon, 2 hours given by Professor Cecilia, who is arriving tomorrow. Oral and poster presentations. Networking opportunities as well will be involved in the event. It aims to foster meaningful conversation, inspire new ideas, promotes connections as well. Highlighting the contributors of women, this event reflects a commitment of excellence, inclusivity, diversity in sensors, empowering the next generation of leaders in sensors technology and biosensing. So, just give you more details about the event, but I would like to invite Professor Antônio Ronaldo de Souza to give some speech for you. Thank you.
Good morning, everyone. As Pedro said, I'm Antonio Souza, Dean of the College of Technological Science at the State University of Santa Catarina. First of all, I'd like to welcome our visitors from Brazil and from other countries that are attending the international workshop on applying biosensor and human in sensors. From different countries, as I said, I knew from Mexico, from Oslo, from Alaska, as well from some Brazilian universities, Fluminense Federal University, University of Brasilia, and Paraná Federal University. Thank you, and everyone that are listen and attending with YouTube. I'd like to thanks in special Dr. Pedro Bertems for the organization of this meeting. I hope we strengthen the relationship between our colleagues from different universities, as I said. Probably most of you know, but Santa Catarina is located in the south of Brazil, and it has around 7 million people, which is one of the lowest populated states in Brazil. From our patterns, Santa Catarina has a good wealth distribution with Gini index of 0.548. In spite of some Brazilian states having 10 years less, our life expectancy is around 71 years. Santa Catarina is the third highest age GE in the country. Nowadays, we have the lowest unemployment rate, and we are the sixth largest economy in Brazil. In the higher education area, Brazil has 192 universities. Of this total, 100 are public. Santa Catarina has 3 public universities like UDESC. UDESC, which has almost 60 years of excellence in teaching, research, and extension. We have multi-campus structure with 12 colleges distributed in 9 cities in the Santa Catarina. In addition, we have also 20 cities with distance learning structure supported by UDESC. We are in, as you know, of course, in Joinville, so we called here UDESC Joinville, that offer both classroom instruction and distance learning. The regional economic focus is industrial and IT, so our courses and programs are in these areas. Here in UDESC Joinville, we have 9 undergraduate courses and 14 graduate courses, 10 masters and 4 doctorates. As I said previously, UDESC Joinville focus on courses that meet the demand for skilled labor in the industrial sector, such as engineering and IT, and also in teaching degrees. Our academic community is composed of 170 faculty members, 88 staff members, 56 lecturers, and around 2,700 students, and also 75 outsourced workers. We are improving a lot. In the last 5 years, the scientific production increased more than 1,000%. There is in UDESC Joinville a balance between recruiting the best student and promoting social inclusion of students come from public schools and the different gender and ethnic minorities. Some keys to our success, if you can allow. First, 4 undergraduate engineer courses in Joinville. We have the first, the second, the third, the 4 undergraduate engineer courses in Joinville. We started approximately 50 years ago. We also have the first 2 undergraduate IT courses in Joinville. First master, first doctorate, second master, second doctorate. So we have a very long contribution for this area. The only HAE in Joinville with permanent undergraduate courses in teaching degree in exact science. UDESC Joinville is the only one university with the permanent courses to teach in these hard areas. In research and innovation area, we have 160 research projects, some of them in partnership with companies and other universities. We also have 30 extension projects in different areas like children's education, elder care, and technical service to industry and government. To final, I hope the events bring to you the best you want technically, and also you can enjoy our city. Have a nice week. Thank you for attention. That's all. Thank you.
Okay, thank you, Professor Antonio. Before officially open, I just give some hints about the schedule of the conference, workshop in that case. We are finishing the event today after the coffee break, okay? We are not going to have the poster session number 1, right? Only one oral session, which is going to start at 10:30, right? And the first lecture today is given by Professor André from the University of Brasilia. The second one is going to give by Professor Serbena from the Federal Technical University of Paraná. That's the one. And a tutorial given by Professor Alexander Sadi Paterno from our department, Electrical Engineering Department here in this campus. And the lunch, everybody is invited to go to the shopping center. We are very close, nearby, few meters walking here. But I also invited, we have a restaurant, university restaurant, close here as well. Okay, thank you, and I can say officially open the event. Thank you very much for coming. I'm very happy. Thank you. So presumably the invite to lecture is going to start in 7 minutes. Is that right? Thank you.
Okay, let's start. So, good morning, everyone. My name is André Nicola. I'm a physician researcher at the University of Brasilia. For those who are not from Brazil, this is the capital city, maybe some more than 1,000 kilometers away from here. I would like to thank very much the organizers for inviting me here to this beautiful city, beautiful state, to talk about this to a group of people with very distinct backgrounds. It's at the same time a bit scary, but incredibly exciting to be here talking to you guys. You see that at first it doesn't make much sense for a physician to be talking here, but I hope this will be a very interesting opportunity to strengthen and start new collaborative work, because I think we can gain a lot by collaborating with you guys. I think you guys can help us solve some of the pressing issues we have. And I'm going to talk about antibodies and infectious diseases, and this is something that I've been working for quite some time, and hopefully it will make sense for you guys why I'm bringing up this subject and why it would be important for biosensors and what sort of solutions we can come up. And also, before starting the talk itself, I just wanted to explain how it came to be that I'm here talking to you guys. I have a long-standing collaboration and a very strong friendship with two researchers here at Santa Catarina who I met when we both work in the US. And we both work on making antibodies to treat diseases, to diagnose diseases, etc. And at some point, they talked to me about a group here from Santa Catarina in Paraná who wanted to write a project along with researchers from Germany. And I thought the idea was excellent. I joined the team. The idea was that this group is making sensors to diagnose diseases. The technology behind the sensors sounds amazing to me, but I really can't explain. Hopefully it will show up in future talks. I know it has something to do with optic fibers and surface plasmon resonance and all sorts of other things I've learned at some point in my career. But then I got invited to be part of this project, both generating reagents that would be used for diagnosing diseases, but also making a bridge with other healthcare researchers who have access to patient samples that would be used to test this new diagnostic technology. So it's a really interesting project gathering healthcare workers with engineers and physicists to try and make new diagnostic technologies that are cheaper, more effective, easier to use. And that's how I came to be here. That's how I met Pedro, Alex, and the other people working on this team. That project, unfortunately, didn't get funded, but hopefully we'll have other opportunities in the future. Right? So I just wanted to start by showing you what I'm going to talk about, the market for this. So the market for diagnostics, for diagnosing laboratories that diagnose human diseases, is worth about $210 billion nowadays. And it's expected to grow a lot throughout the next years. So this is a huge market. There's a whole lot of money on that. And there are several thousands of different tests using many different types of technology, but one key piece of technology in this market is antibodies. We use them as probes, we use them to diagnose disease. And the market for antibodies in diagnosis is estimated to be around $20 to $30 billion a year. So it's a little more than 10% of the entire diagnostics market just to make one of the reagents that is used on these kits. So this is what I'm going to talk about. And this is a huge market hopefully I would like to show you on the first part of this talk what the antibodies are and why are they so useful that they're a huge chunk of this market. But also I wanted to talk to you about some ideas I have for new strategies to diagnose infectious diseases and why I think we physicians and biomedical researchers cannot solve this problem by ourselves, but I think by collaborating with physicists and engineers and people in very different areas, I think we can solve some pretty important issues. So this is what I'm planning to talk to you about today. First, talk about antibodies, which I love, and hopefully you'll be interested in the technology as well. But then show you an issue that I think needs solving, and I'd be happy to talk to you guys about ideas you have for solving that. Okay? And just a parenthesis here, I'm going to be talking a lot about diagnosing human diseases, but this makes, the idea is the same for diagnosing animal diseases and diagnosing a lot of other different stuff. But this also makes sense outside the biomedical area altogether. I once took a course in the US on antibodies, and there was a researcher there from the MIT, and she used antibody technologies to diagnose small fissures in helicopter rotors that were better visualized with antibodies than any other technology, and the US military was using that technology. So you can apply pretty much everything I'm going to talk about here today in infectious diseases, but you can apply that in all sorts of different areas. And hopefully, again, you can get great ideas from what I'm going to show, and maybe you can use antibody technologies to something in your specific area of expertise after hearing about them today. Okay. So antibodies are a pretty important part of the history of biomedical sciences in the last 100 to 150 years. The very first Nobel Prize in medicine in 1901 was given to the researchers who, well, one of the researchers, Emil von Behring from Germany, who discovered antibodies. He discovered that with other researchers, but he was the one who got the Nobel Prize. And here's a list of Nobel Prizes given throughout the following century to either specifically to antibodies, like people who discovered how the genetics of antibodies are made, or the structure of antibodies, but also to different technologies that use antibodies, like the most recent one from 2018, some drugs that are based on antibody technology that are being used to treat cancer. So there's a whole lot of Nobel Prizes, and there's a whole lot of biology that runs around antibodies. This is the basic structure of an antibody, and this is going to be interesting to understand why they're so useful. So these are proteins. They are huge molecules. The molecular weight is around 160,000, so almost 1,000 times larger than a water molecule. And they have 4 different polypeptides, 2 copies of a light chain, 2 copies of a heavy chain. So these are 4 different peptides, 4 different proteins that are bound together covalently to form this Y-shaped structure. This is the most common antibody structure. There are other formats for antibodies on different animals, or even within humans there are different shapes. But this is by far the most common, the one we call IgG. It's the 2/3 of the antibodies on our body are shaped like this. And they have on this edge here a variable region, and the rest of the antibody is constant. This means that the constant region doesn't change a lot between antibodies. The variable region changes a lot. So we have several million different types of antibodies in our body. They all differ here, but they are very similar on the rest of the antibody. And these 2 regions are incredibly important for antibody biology. The constant part is really important in immunology, which is what I teach, but I'm not going to talk a lot about it today. What matters for antibodies as technological tools is this variable region here at the end. And just some lingo that so you guys can follow up what I'm talking about. When I mention antigen, antigen is the target to which an antibody binds. Epitope is the part of the antigen that is recognized by the antibody. So you can have a very large antigen, but just a specific section of it is recognized by the antibody. That's called the epitope. And conversely, the paratope is the part of the antibody that binds to the antigen. And the paratope is here at the very edge of the variable domains. And another important concept here is affinity, is the strength of the bond between antibodies and antigens. The higher the affinity, the stronger the antibody is binding to and recognizing the antigen. So when you look at the antibodies, they have these domains here. So the heavy chain has 4 domains, the light chain has 2 domains. Each one of these domains has basically this shape here. It's like, they're called immunoglobulins because of this. They form sort of a globule with some parts of the protein sticking on the surface. And you get these, the edges of the light chain and the heavy chain, and they form the paratope here. So this is schematics, this is actual biophysical data, the structure of the atoms that form the paratope on 4 different antibodies. And hopefully you can see here that there's a small red thing here, that's the antigen. So here's a tiny antigen, here's a much larger antigen. And these antibodies, the very tip, the variable part, has different amino acid sequences because they come from different genes. And these different amino acid sequences mean that they have different 3-dimensional structures. You can see here on a side view, this one has a protrusion. This one has sort of a valley or cup shaped surface. This one has a very tiny pocket. So the 3D structure differs on the very edge of the variable domain. And this is what makes antibodies able to recognize different antigens. It's something that we learn in first year in any biological course, is the key and lock principle. The idea is that molecules interact, biomolecules interact if they have 3-dimensional structures that are complementary, like a key fits in a specific lock, or a glove fits in a hand. If you have complementary 3-dimensional structures, the molecules will bind to one another. Right? And how does the body use antibodies? And this is interesting to understand how they're useful as technologies. So antibodies are made by cells we call B cells, and these B cells are circulating through our body. They're made somewhere in the body, and they keep circulating for years. And we have millions of different B cells. Each one of the B cells has several copies of antibodies on the surface. But each one of the B cells has a different antibody. And the difference between them is the 3-dimensional structure of the variable domain. So these B cells, they're just going around the body, circulating, looking for antigen. Whenever an antigen enters our body, let's say an infectious a microbe that causes infectious disease, this microbe or this antigen is going to start interacting with different B cells. And at some point, a B cell whose receptor, whose antibody, has a 3-dimensional structure that is complementary to the 3-dimensional structure of the antigen, will recognize this antigen. And once this B cell recognizes the antigen, it goes through a lot of cell divisions. We call this clonal expansion. It generates several thousands or millions of copies of the same cell, and these cells start producing antibodies. And these antibodies then are secreted from the B cell, and they go on to multiple parts of the body to have a huge number of different immune functions. And this is basically two-hour lectures of two-hour immunology lectures condensed in 2 minutes for you guys. So we spent a lot of time talking about what antibodies do. But here it just suffices to say that they have a lot of different roles in immune responses to bacteria, viruses, and cancers, and other different things. Another interesting concept here is the concept of specificity and cross-reactivity. Specificity is the ability that antibodies have to recognize one antigen but not a different antigen. And they do this because they have differences in shape. But an antibody that is made by our bodies to recognize a specific antigen can recognize a different antigen with a similar three-dimensional shape. So you can have here the antigen, the epitope, which is a part of the antigen that's recognized by the antibody. But this antibody can recognize an identical epitope on a different antigen, or can recognize a similar epitope on a different antigen. So this is cross-reactivity. It has a lot of different biological roles. Since I'm going to be talking, as I mentioned, about diagnosing human diseases, cross-reactivity is something that is a problem. So you can have a test against dengue viruses detecting Zika virus infection, for instance, because the viruses are very similar. Right? So this has specific biological roles. In diagnosis, this is something that we try to avoid as much as we can. Right? So the immune response, of which antibodies are part of, the immune response has to be pretty specific. And this is something where antibodies are great, and that's why they're really useful tools for the immune system. These two patients here have infectious diseases. This one is leprosy, this one is HIV, plus AIDS plus tuberculosis. But the damage to the skin here and the damage to the neck and the liver, from which 2 liters of pus were removed, the damage here is done by the immune system, not by the microbes. So in some infectious diseases, the immune system responds to the infectious microbe, but also that response actually damages the host. And this is a problem. So it's something that biology needs to deal with, and the best way to deal with that is to make specific immune responses. So the more specific the immune response is, the more it harms the bug, the microbe, or the tumor cell, the less it harms the host, the person who's harboring that microbe or the tumor. And that's why antibodies are so useful. They're incredibly specific because of that three-dimensional complementarity of sequences. And this is, the specificity is a hallmark of antibodies in the immune response. By the way, these are not caused by antibodies. This damage is caused by other parts of the immune system that are less specific. But this biological characteristic of specificity is what makes antibodies great tools for diagnostics. They are incredibly specific and they have very high affinity. So specificity means we can have antibodies to detect different diseases. For instance, with one single type of virus, flu virus, the one that causes influenza, grippe, we can have antibodies that separate a single virus species in 240 different types. So you can have antibodies that are not only able to recognize a single species, but to separate a single species of living beings into 240 different groups. That's how specific they are. So they make really great diagnostic tools. But also they bind with a very high affinity, which makes tests using antibodies really sensitive. You can detect very small amounts of whatever it is you're trying to detect, so you can detect a disease pretty early on the course. So these biological, the whole specificity thing that is important for the immune response is what makes antibodies great tools. And they make great tools for diagnosing diseases, which is the focus of my talk, but they also make really great tools to work on basically anything else. As the work I mentioned to you guys about the helicopter rotors, she made an antibody that bound to fractures in metals. So whenever the metal got fractured, there were some electrons that were, I don't remember the physics behind it anymore, but something changed on the electron pattern on the metal when there was a rupture that was on an atomic scale. And she made an antibody that was able to detect these atomic scale fractures. So you can use this on helicopter rotors or maybe on fish that are swimming in Alaska or any other ideas you guys can think of, you can use antibodies to develop new technologies. So how do we make antibodies? The first antibodies were what we call polyclonal. You get an antigen, you inject it into an animal. Here, this figure shows a rabbit, but you can use goats, mice, horses, all sorts of different animals. You inject, the animal will recognize this as being foreign and will make different antibodies. So these antibodies are made by different cells. Each cell is going to expand, as I mentioned to you earlier, and form clones. So this is why we call these antibodies polyclonal. They're made by different B cells, and you can see here that different B cells have different shaped receptors. So you end up with a mixture of antibodies, and they have different structures. So they're able to recognize several different epitopes on the same antigen. These are polyclonal antibodies. You inject them into animals, and we have huge factories to make these antibodies in animals because they're incredibly useful for diagnostics. The other technology is, the other way we make antibodies is we use hybridoma or phage display. These are two different technologies, both of which were rewarded by Nobel Prizes, and these make what we call monoclonal antibodies. They come from a single B cell, so every single antibody has exactly the same three-dimensional structure. And the basic idea behind the most used technology is you get B cells, either from a person that had the disease or from an animal. These B cells produce antibodies, but once you take them out of the body of a human being or an animal, they die within 1 or 2 weeks. So you can't have them produce an antibody on the lab or on a factory. You fuse these cells with another cell, which is a cancer. It cannot produce antibodies, but it can replicate in the laboratory. We call this an immortal cell. Once you fuse these two cells, and we call these cells a hybridoma, OMA, if you remember, you must have heard it at some point, this is something we use for cancers. So this is a hybrid cancer. This hybridoma cell inherits from this parent the ability of producing antibodies, from this parent the ability to replicate in the lab. So we get from this one cell, we start multiplying, and then we end up with several trillion cells in a 10,000 liter tank, and then you can produce kilograms of antibodies. And the antibodies are all exactly the same. They have exactly the same three-dimensional structure. So this takes a lot longer to make than this, but this is really variable. You need to keep injecting different animals. Sometimes the animal responds, sometimes the animal doesn't. There are ethical problems here in using animals, making whole industries of animals that are used to make antibodies. But again, there are pros and cons for the polyclonal antibodies and the monoclonal antibodies. But these are two very important technologies that we use to generate the antibodies that go into the diagnostic chips or diagnostic tests. Right? How do we use these antibodies? I'm going to show you guys two examples here of technologies that are very widely used in medicine that depend on antibodies to diagnose diseases. The first one is the enzyme-linked immunosorbent assay, or we always use the acronym ELISA, or ELISA in Portuguese. It's not a female person, it's a specific test. The idea is, first we get a plastic plate. This is a picture of the plate. It's pretty much this size here. And it has 96 holes excavated in it, and it's a plastic material to which proteins bind. So you just put antibodies there, the antibodies bind to the surface of the plate, and then you add something, let's say you add a person's blood, and this antibody, let's say it detects, it binds to SARS-CoV-2 protein, a protein on the coronavirus that causes COVID-19. Then the coronavirus will bind to the antibody. You add a second antibody that binds to a different epitope, a different part of the virus, and then you have this sandwich here. One antibody recognizing the virus, and the virus being recognized by a second antibody. And this antibody here is covalently bound to an enzyme. This enzyme catalyzes a reaction that transforms a colorless substance into a substance that has color. So as you can see here, there are some wells that have no blue color, some wells that have a very light blue color, some wells that have very strong blue color. The more color there is, the more enzyme there was on that well. More enzyme means more antibody means more antigen. So in this case, if this was a SARS-CoV-2 ELISA test, the bluer the well, the more virus there was on the person's respiratory fluid or blood or whatever it is the sample you put. So you stick the antibody to a solid surface, and this is important, I guess, from the engineering point of view on how you guys can use the technologies you work on to make diagnostic tests. So you stick the antibody to a solid surface, and then you need to detect the antibody somehow. In this case, we detect with a colorimetric enzymatic reaction, but you can detect with light, you can detect with fluorescence. Hopefully we can detect with impedance changes or other different types of measurements that will detect this, the binding between antibody and antigen. Right? Another example is the lateral flow assay. It's the one that, if you've ever done a pregnancy test, a home pregnancy test, or if you've done a test for COVID-19 at home during the pandemic, this is the technology you use. You can either get a strip of paper, a naked strip of paper, or you can get this strip of paper packaged inside a plastic contraption like this or like this one. This is more similar to the ones we had during COVID-19. Have you guys ever done this at home? You know, you just stick something in your nose, you sneeze afterwards, and then you poke it into liquid, put some drops. That's the sort of test I'm talking about. So the liquid in which you dissolve your sample has a bunch of colored balls, and to these balls we attach covalently one antibody. So whenever you put the patient's samples, this antibody will come in contact with the sample, and if there is the antigen, you have the antibody binding to the antibody will bind to the antigen that's present on the sample. Let's say if this is a COVID-19 test, it binds to the virus. And then the liquid starts migrating through capillary action, and it gets to a bend here in which we bind antibody to the paper. And this antibody recognizes a different part of the SARS-CoV-2 virus. So if there is virus on the patient sample, the virus will make this sandwich with the antibody that's bound to the paper and the antibody that's bound to the colored balls. And these colored balls will start precipitating on this line. If there is no virus, the balls do not bind to this test line and they keep migrating until they reach the control line. So you have a negative test here, just a control line. You have a positive test here in which the sandwich is formed because there is antigen. So if the antibodies if this pair of antibodies is made to recognize SARS-CoV-2, you can then detect the presence of virus on the substance. And this is something that you don't need fancy machines, you don't need trained personnel, you just need to know how to read to make this test. You can make it at home, you can store this at room temperature, so these are incredibly useful tests. This is what we call point of care tests. You can do it at the bedside, you can do it at home. This one requires equipment. It's not that expensive, but it's about $2,000, the equipment to read these plates, and it maybe costs $100 or so per plate to do the test. This one is pretty cheap. You can get this test for $2 for some diseases, and again, you just need to know how to read to make. So this is hopefully the type of assays that we want going into clinic in the future. So how do we create a new test from scratch? The first step is we need to choose a good antigen. So we need to figure out what are we going to use to detect. And again, if you're thinking about a bacterial disease, bacteria have several thousand different antigens. So you need to figure out which one of those. You can either get the entire bacterial cell and just put it on a blender and make a soup out of it and use that as antigen. That was how things were done 100 years ago. We don't do that quite often anymore because there's a whole lot of issues with using whole microorganisms. you can, instead of getting the entire microorganism, just getting one protein purified from that microorganism. Or if you want to be even more precise, you can get just a fragment of one of the antigens that's present on the bacteria. This makes for a test that is way better because you're just using a small fragment which eliminates other things that could be similar between different bacteria or different viruses. So this is preferable, but you need to choose which piece of which one of the several thousands of proteins are present on a different microorganism. And this is empirical. You just you test a whole bunch of different proteins and you end up with one that actually works. So this could take maybe a couple of weeks, maybe a couple of decades. It's empirical. You have to keep trying until you find one that actually works. Once you find an antigen, you need to make antibodies. And then you can make antibodies in animals, those polyclonal antibodies. You can make monoclonal antibodies. And the idea is you generate dozens or several hundreds of different types of antibodies. And again, it's empirical work. You keep testing all the different antibodies to find one or a pair of antibodies that work for what you want. And again, empirical work could take 2 weeks to 2 decades, depending on the antigen you're working on. After you have the antigen, you have the antibody, you need to start doing other empirical work to figure out which assay technology, ELISA, lateral flow assay, chemiluminescent assay, which one works best. You need to test different concentrations of antibodies, different concentrations of the reactions. And ideally, you want to come up with something that is easy to use, is cheap, is very sensitive and specific. I'm going to talk about this in pretty soon. And this is actually pretty hard to come by. The ideal test is really hard to come by. Pretty much every test we have on the market has some drawback or another, right? But this is, again, as we were talking on the project about getting a specific device to make tests, this is what we're aiming at, getting something cheap, easy to use, that could be used on the point of care instead of a diagnostic laboratory, and that someone who knows how to read can perform. So the whole way we do diagnostics nowadays is we have one test per disease. So you have one test for influenza, one test for dengue, one test for Zika virus. Maybe you have 2 or 3 different tests for a disease. For hepatitis B, we have 6 different tests for the same virus. And they all show slightly different aspects of the disease. But again, we tend to do one or a few tests per disease. And I think that's a problem. And here's where I come to what my idea is, my suggestion is for, I'm going to present you guys a problem, and hopefully we can work together to figure out a solution for that. I think we should make several tests for each disease. Instead of spending several years looking for the perfect antigen, the perfect antibody pairs to develop one test for one disease, we should be working on getting several antibodies, several antigens to make several tests for each single disease. And there are 2 reasons for that. The first one has a lot to do with statistics and Bayes' theorem, which I have not learned in medical school. I suppose a lot of you might have learned in engineering or physics. It's apparently the most important theorem in statistics. And there's a really interesting paper arguing that clinicians, physicians, people who treat patients are natural Bayesian machines, that we use the Bayes theorem. I only figured that out 20 years after I graduated from medical school, or 15 years after graduating. But the idea here is you have people that have a disease or do not have a disease, and if you test them, if you run a lab test, the test can come positive or negative. No test is 100% perfect. So you always have some people who do have the disease but have false negative results. You always have people who do not have the disease but have false positive results. This always happens. The tests are never 100% perfect. So we talk about sensitivity, which is the proportion of people with the disorder who have a true positive result. And we talk about specificity, which is a proportion of people who have negative results among all of those who do not have the disease. So specificity and sensitivity are characteristics of each specific test. And here's an example of how one of these tests would work, a test to diagnose HIV infection. And these are actual numbers you can get from the kits. These are the closest to perfect we can get with a diagnostic test. Sensitivity of 99%, specificity of 99%. And here I'm just running this test in 100,000 people, 40% of which have HIV infection. So you have 100,000, 40% have the disease, so you get to 40,000 here, 60,000 here. From the sensitivity of 99%, you end up with 39,600 people who have the disease have a positive test. From specificity, you come up to 59,400 of those people who do not have the disease, have a negative test. So these are characteristics of a test. This doesn't change between different people, different situations. But this doesn't make much sense when a physician's testing patients. When you run a test, you get a patient who you think have a disease, you run a test, and then you want to know if the test is positive, does the person really have the disease? Or if the test is negative, does the person really does not have the disease? So here we need to talk about positive and negative predictive factors. So positive predictive factor is you divide this by the sum of this line here. So 39,000 divided by 40,000 means if the person you ordered the test has a positive test, the chances of the person actually having the disease are 98.5%, which is pretty good. A very small chance you're going to say to someone you have HIV and the person does not have the disease. Also, if you have a negative test, you can be 99.3% sure that when you tell the person you do not have HIV, that the person actually does not have HIV. So these are the things that actually matter when you have a patient in front of you, not sensitivity and specificity. These are the things that matter. And this looks like a pretty good test, right? But then you go to a different situation, and we only change the prevalence. This means that out of the 100,000 people, only 0.01% have the disease. And you do the math here all over again, and then you calculate the predictive factors, and you end up with 10 divided by 1,010. That means that a person is in this situation, comes to your office, imagine you're a physician, has a positive HIV test. You tell the person you have HIV infection. You have 1% chance of being right. 99% chance of telling the person they have HIV and they don't. So the same test that looked pretty amazing in this other situation is pretty lousy. You have 99% chance of making a mistake when you tell the patient they're infected. And what changed here is the prevalence. So prevalence means a lot. How do we change prevalence? How do we work with tests on a situation of high prevalence? And this is where the Bayes theorem comes. This is the formula of the Bayes theorem in case someone wants to remember it from undergraduate school. How do we do that? And this is a paper that was published early in the pandemic, and this shows symptoms, different signs and symptoms of COVID-19 in people. So 83% of people with COVID-19 have fever, 82% have cough. So what we do as physicians is you start with a chance of, let's say 1% the person has COVID. That's how many people in the population have COVID at a specific time point. And then you ask the person, do you have fever? If the patient has fever, you increase from 1% to 1.8 or 2% the chance that the person has fever. And then you make another question, are you coughing? And each time you make a question, you put those numbers back into the Bayes theorem equation, and you keep increasing the chances the person has COVID-19. So we do this for several different questions, several different tests, and at the end we end up with a patient that has 80-90% chance of having COVID. And then we run the test. So we're running after we ask questions and do the physical exam, we can have tests that actually work. So you see that we're making this in several different steps. And I think this would be great for diagnostic tests. If we have several different tests for the same disease, we're making exactly the same thing. One test increases the chances of the other tests being more precise. And this is one of the reasons why I think we should do sequential tests. We should be doing several tests for the same disease. The other reason is something that is even broader, and it's something that is a subject I like a lot as someone who teaches immunology and microbiology. When you think about the immune system, what we learn in school and what most people say is that the immune system is a system that defends us from our enemies, defends us from bacteria, viruses, whatever. So here's a definition on a paper by immunologists. It's a system that fights pathogens and destroys microorganisms. Here's a dictionary. Again, it uses the word fight. So we basically see our interaction with bacteria, viruses, fungi, protozoa, helminths, or worms. We see our immune system protecting us from them, and their goal is to actually kill us or feed from us. So they're parasites, they're trying to kill us. This is the germ theory of disease that started. It's the result of a few hundred years of research being done. And it's one of the most important, most successful models in all of human science. Right? So how do we deal with infectious diseases? We either prevent contact with microorganisms, or we kill the microorganisms. And this is the basic idea of treating infectious diseases. Or you prevent, or you kill whatever gets into your body using antibiotics or whatever other tools you have. But this is a problem. Not all microbes are our enemies. Actually, most microbes are not our enemies. And this is the example of bacteria that are present on the vaginal mucosa. These bacteria make acid. They feed on sugar that woman's body gives them. They make acid. This lowers the pH and protects the vaginal mucosa from infections. If the person washes off the bacteria, the pH raises, and then fungi start multiplying, and they cause disease. So this is a bacterium that is actually protective in our body. And most bacteria are actually beneficial for us. So if you just think that we need to kill the bugs, we end up with problems like antimicrobial resistance, which is expected to kill about 10 million people per year in 2050. Or we end up with a disease called pseudomembranous colitis. This is an intestine that is full of bacteria, and this is a disease that's causing people by giving them antibiotics that kill most of the bacteria in the gut. This one bacterium does not die from the antibiotics. It starts multiplying because there's no competition. It generates fever, diarrhea, and it kills a lot of people in the ICU. So this is a disease that we cause in people because we're trying to kill bugs, because we think our goal is to kill microbes. So don't get me wrong, killing microbes is good. This is one of the largest revolutions in human history. This is a graph showing life expectancy. The whole germ theory of disease was made at the late 19th and early 20th century, which is around here. So you see different countries, the life expectancy was around 30 to 40 years before the germ theory of disease, 70 to 80 years after the germ theory of disease. Here we have child mortality. 200 years ago, about 40% of the children died before they were 5 years of age. Nowadays, we have less than 5% of kids dying. And there was a lot of different technologies, a lot went on in these 200 years, but a really important thing here was the germ theory of diseases. And we were able to treat several different infectious diseases. But this idea of killing the bug is actually not perfect. And as I mentioned to you, there are problems with that. And we sort of, I feel we've hit a dead end. We can't move forward a lot more by just thinking about killing the bugs. We've done pretty much everything we can to kill the bugs, and we still have infectious diseases going on. So how do I think we should work on this? There's this pretty interesting graph from two infectious diseases physicians from the US. It shows here on the x-axis how strongly the host responds, how we would respond to a microbe. The further you go to the right, the stronger the response is. And here is damage. The further up you go, the more damage there is. Further down you go here, on the negative part of the y-axis, the more benefit. So you can get benefit from interacting with microbes. the idea that they propose is that almost every infectious disease has this sort of parable here. If the immune system, or if the host responds too little, you have a lot of damage, and you can even have death. This is what happens in people with AIDS, for instance, or in people who have transplant, and you have to give them something to decrease their immune system. The immune system doesn't work really well, microbes get in, they multiply, they kill the person. On the other side here, you have things like COVID-19, for instance. What kills people with COVID-19, and we've had several million deaths in the past few years, is not the virus, it's the inflammatory response against the virus. So it's the immune system that reacts a lot stronger to the virus, and the damage the immune system causes to the virus ends up damaging the host as well. So you can have disease by having too little immune response, you can have disease by having too much immune response. And the interesting thing here is that we can, if you think of diseases instead of being caused by our enemies, the microbes that we need to kill, if you think about diseases that they can be caused by the microbe or they can be caused by the immune system, you come up with a different way to treat disease. You can modulate the immune response. So if the person's here, you give the person something to decrease inflammation, you decrease damage. If the person's here, you give them something to increase their immune system, and then you decrease damage. So this is, instead of targeting the bug, the microbe, you target the host to treat infectious diseases. One of the great challenges in doing this is that we don't have tools to figure out when a person comes right in front of you in a hospital if they're here or they're here. We don't know how to diagnose that. So we can't really give them something to modulate the immune response because we don't have tests to figure out where the person is on the curve. And that's where I think the new technologies in tests could come. The immune response is incredibly complex. We can't figure out this by just testing one specific molecule. We'll probably need to test dozens or hundreds of different molecules to understand where the person is on the curve, here or here. And that's why I think a technology that can use multiple antibodies to detect multiple antigens can be incredibly important in diagnosing disease. If we can, instead of looking for bugs, looking for host proteins to understand the immune system, let's say you have a chip that has several hundred different substances and you're right at the patient's bedside, you put a drop of blood on this chip. 15 minutes later, a result comes and says the patient is here. You use a drug to decrease the immune response. Or the patient's here, you use a drug to increase the immune response. We don't have the technology to do these tests. And that's where I think the collaboration between healthcare workers and engineers and physicists could come and could revolutionize diagnostics, infectious disease diagnostics. And with that, I would like to acknowledge funding for the work that my lab does, even though I didn't show any primary data here today, and acknowledge all of you who came here and the meeting organizers once again. Thank you.
So we have a couple of minutes for questions. Anyone?
That was an excellent talk. I enjoyed it. I was wondering why have people not been developing antibiotics or something that are specific at the antigen level? Like if you had something that, because antibiotics are there's broad ones and there's specific ones, but they're certainly not specific at the antigen level.
Nope, they're not specific, and that's the first antibiotics that came into the market were reasonably specific, and throughout the decades, the whole pharmaceutical industry moved towards less specific antibiotics. The idea is that the more broad the spectrum, the more types of bugs it kills, the more you sell the drug, because it's going to be used on more patients. And also for a physician, figuring out which exact microorganism is infecting a person, it's hard. And it takes time because the tests take a long time. Sometimes tests can take 2 months to come back, the results. So if you have something that kills pretty much every bacterium, you don't need to wait for the test results. You just give that and you're going to kill every single bacterium, so you treat the disease. The big problem is then that you're killing so many bacteria that you end up with antimicrobial resistance and pseudomembranous colitis and a whole range of issues. But it's market forces that drove the antibiotics towards having ever larger spectrum. And that's a pretty big problem in my opinion.
Mm-hmm. But you can use antigens to detect at a very specific level.
Yeah.
But why couldn't you have, even like there's like antivirals kind of don't exist that,
Yeah.
you know, and so why, I wonder why there hasn't been, at the same level of detection, how come there isn't something to combat antigen?
We have some reasonably new technologies. Those antibodies I mentioned that were the monoclonal antibodies, the technologies from the late 1970s. On the early 1980s, antibodies came to the clinic to treat cancer. And we have several dozens of antibodies to treat cancer. And they're incredibly specific. They target a specific type of breast cancer. So if you're using one to treat breast cancer, it's not every single type of breast cancer that would work. They're incredibly specific. We have a lot of those for cancer. For infectious diseases, until 5 years ago, we had one. Now we have about a dozen on the market, and the antibodies are really specific. That's one of the areas my lab works, actually, trying to develop new technologies to treat infectious diseases using antibodies. But this is something that's brand new. Throughout history, antibiotics tend to be very non-specific.
So if you had an antibody that bound to an antigen, then if you had enough of those bound, then could it make the antigen ineffective? Like
Yeah,
like you're
that's the
looking
idea.
at pollen or
That's
something
the idea.
like that.
It binds to a virus, it stops the virus from interacting with the host cell, and thus preventing infection. We had 5 drugs on the market for this. Do you guys remember early in 2020 when Donald Trump got COVID and he was treated with an experimental drug? That was a bunch of antibodies mixed up together, and they bind to the virus and stop the virus from interacting with the cell. That's exactly how antiviral antibodies work.
Ah, perfect. Thank you for that explanation.
You're welcome.
Any further question?
Thank you for a very interesting and good talk. Could you say a little bit more about how you think that physicists and engineers could contribute in this area?
So the main test we use nowadays is ELISA. And the other tests are reasonably similar. We get a solid surface, we stick antibodies to it, and then we detect antigen-antibody binding. But these plates are large. This is the size of an ELISA plate in which you can test basically 90 samples. If we were to make tests to detect several hundred different molecules per patient, you would need several of these plates per patient, which makes the tests really not feasible with the technology we have. But if you guys were to come up with some sort of chip or fiber optics, something that could make very tiny regions for antibodies to be bound, and let's say on a chip this size you can make 500 or 1,000 different tests, then we could use several hundreds of different antibodies to detect different substances in a single chip, in a single test, and we could use this to understand the immune response and make host-directed therapy a reality. Because we do have some tools to change the immune system. We just don't have tools to figure out where the patient is on that curve. If the patient needs to have their immune system decreased or increased, we don't have tests. So coming up with those tests would be great. We have the biologists on one hand to get the antibodies, but we need a platform in which these antibodies can detect the antigen, which is cheap, small, easy to use. That's where I think the challenge comes. We can't do that. I hope you guys can.
Okay, thank you very much.
Thank you for your talk. It was really nice. When you showed the antigen, the Y shape,
The antibody.
yeah, the antigen.
No, the antibody is Y-shaped.
The antibody.
The antigen
Okay.
can have all sorts of different shapes.
Okay, sorry. When you do the ELISA test, you just put a droplet, and you don't know the orientation of that
Yeah,
Y, right?
you put the antibodies there, they bind to the plastic, 98% of them bind in some way that they're destroyed, 2% of them bind on a way, an orientation that actually works.
Okay, that was my question. If
Yep.
there are ways to improve this orientation, and because for the sensing device, this is really important, right?
Yep, I don't know if there are ways. I hope you guys can have some interesting ideas. The way we do it in our labs is we just throw it all there, and we know that most of the antibodies are going to get denatured. They're going to change their structure to when they bind to plastic and they don't work anymore. If you have any ideas to make that better, I'm all ears. This would make tests way better than they are nowadays.
Okay, thank you.
Any further question? Oops. No, you have one.
Thank you for this excellent talk. So I think this problem, multiplex detection of antigens, can be solved using microarray technology.
Sure.
But and my question is, do you have a pool of targets to be detected already in your mind?
Oh, that's exactly the point. In my mind, I do. In the real world, no. We have several different assays on the market. It could be just a matter of combining them all to a chip. But I think we'll need more than what's on the market. This is something that, this whole idea of host-directed therapy, I've just come from a meeting in Washington, D.C. last week. It's not just me, there's a whole bunch of other people working on that. And I tell my students, I hope by the time I retire, physicians will not just be applied microbiologists. We'll try to figure out if it's a bacterium, a virus, and then we use something to kill whatever microorganism. But I hope people will be also applied immunologists. This is not the reality today. But this is where I want to come in 20 years or so when I'm about to retire. So to answer your question, we do not have them yet. There's a lot in mine and other researchers' minds, but it's not real as of today.
Okay, thank you. So maybe we can discuss something else.
Sure.
So we have time for the last question. In 2 minutes, coffee break is going to serve it. Is it answered? Okay, so thank you very much. And
Thank you.
everybody's welcome for the breakfast. Thank you. So we are starting the first oral section of the workshop, and the first presentation will be given by Pablo Dutra da Silva. And the title of the article is Comparison between Switched CMOS Current Source with Enhanced Howland Circuit Spice Simulation. That's the first one. And I would just like to ask to call Lucas Alves de Lima. He's here. Ana Paula de Oliveira Souza. Rosa Amália Firman Dutra. This was supposed to be the second presentation of this morning. If they are not here, no, so I'm afraid you have going to have only one, and then after that we are finished for tomorrow morning, and we are ready for lunch further on. Okay, thank you.
Good morning for everyone. My name is Pablo, and I will present you comparisons between switched CMOS current source with the Janssen-Howland circuit, SPICE simulations. Well, wearables are becoming very popular for some applications nowadays. We can see it, lifestyle tracking, performance tracking for athletes, and research for health and monitoring and health problem detection. And is supposed to be in the future used for medical instruments as well. But wearables and the non-invasive or some non-invasive detection of health problems have some technological requirements for I will cite only the circuit requirement for this kind of device. Small volume, low voltage supply, and low power consumption, and high precision. To achieve this kind of requirements in circuits is mandatory the use of microelectronics or integrated circuit designs in order to minimize the volume, the low power consumption, and low voltage for this kind of circuit. We can see that this picture for open wearable device, you can verify the small volume circuits, electronic circuits on this kind of device. So, a wearable that uses the technique of bioimpedance spectroscopy is becoming commonly used, the time domain technique, which is a technique that injects signal, which is in sometimes a pulse or a train of pulses, the binary pulses, randomly distributed between a time interval. It will result in a signal with a whole bandwidth to interest for the test, and is becoming popular for its fast response versus the frequency domain technique, which is a sweep of frequency of a sine wave. And one of the most important circuits for this kind of, to apply this kind of technique is the current drivers. The most used is the Holland current drive, or Janssen Holland current drive, that uses linear circuits, usually op-amps. The output impedance of this current driver is highly dependent on resistance matching and open loop gain. The current output highly dependent to the voltage, the signal voltage generated, and then this generated of voltage could be very precise. It requires high performance op-amps because it's for time domain analysis. We have a pulse of train of pulses, and the transitions of the levels of that pulses should be fast. And low output voltage swing. In comparison with a switched technique that we call switching CMOS current source. It's based in H-bridge switching structure. We will see that it is simple and fast, permits low voltage and low power supply, is exclusive to integrated circuit and time domain bioimpedance spectroscopy. High output voltage swing, high load capability, and usable for tetrapolar analysis. The topologies under comparison, we have the Janssen Holland current source. It's this represented with this diagram. There are some requirements to make this relation of a simple Ohm's law true for this circuit. And these requirements are a perfect matching in R4 and R5, and a matching of R3 equal R2 plus R1. These are the conditions to make this relation true, ideally. So this is the equation, the project for equations. We used output current specification of 250 microamps, used supply voltage and the high level of the pulses of 3.3 volts, and a reference voltage of 1.55 voltage. With that, with those values, we can get that values of components. The capacitors are decoupling capacitors. They should be as big as possible. And we used a micro model of OPA 2355, which is high performance operational amplifiers. The second, and our purpose, is the switching CMOS current source. In this diagram, you can see the two main parts of this circuit. The first one is the current mirror. It's a high impedance current mirror. And we have a CMOS H-bridge constructed with CMOS inverters. For the first design of this structure, for the exploration to make this comparison, we used the same current specification, the output current of 255 microamps, and we took care with the VDS set of those transistors M8 and M7 to improve the output voltage swing of this current driver. We can design the transistors dimensions using the technological parameters of 120 nanometers CMOS technology. And we got those dimensions that was used to the SPICE simulation. The transistors of the H-bridge was designed to be fast enough for the technology. It's designed to be the most small as possible. Here, I'll present you in that diagram the electrical schematic for the load. We use a distribution of electrodes for 4 electrodes in the tetrapolar scheme and used for a representation of the tissue under analysis. This circuit, this electrode circuit with a resistor of 1 kilo ohm and capacitor of 1 nanofarad. In this arrangement, when we have the electrodes 1 and 2 responsible to the current injection, and the electrodes 3 and 4 responsible for the voltage measurement, we got that the Z measured is the individual impedance over 4. This condition is true when that branches are equal. So, for the bandwidth of interest for our study, we have a calculated impedance of 403 approximately ohms for 100 kilohertz. That is the beginning of our bandwidth, and a Z measured at 1 megahertz of 284 ohms approximately. So now we can verify in that 4 graphics the comparisons of a DC analysis for both circuits. The first one, the A graphic, we can see the output current versus the output voltage. For this analysis, the circuit was set up to a condition that will offer the specified output current and a sweep in a voltage source in the load electrodes is done. We can verify in this graph that the specified current is deviated of the Howland in a output voltage around 1.6 volts. And for the CMOS current source, the deviation starts approximately at 2.5 volts, which gives us a wider range of voltage, the output voltage, valid to maintain the output, the specified output current. There's a remarkable fact that should be talked is for the CMOS current source. This graph is mirrored for the negative potentials as well. So we have an output voltage swing of plus or minus 2.5 voltage. In comparison with the Howland source, that is an output voltage swing of 1.6 voltage only. This same conclusion we got in the C graphic with this calculation of the error between the simulated current and the specified current. We can see the deviation in that 1.6 volts for Howland and 2.5 voltage for the CMOS driver. And about the output impedance of that drivers. We can see it in the B graphic. When we verify a high output impedance around 20 mega ohms for the CMOS driver till 2 or 2.5 voltage, and high impedance of the more than 80 mega ohms for the Howland source. This result. There's a fact. We said the output impedance of the Howland is highly dependent of matching for the resistors. So the simulation don't consider this mismatch. So we are considering the resistor matched. So again, in real design, the tendency is that the impedance will low this value. And other thing interesting for the CMOS is that the predominant impedance, output impedance is of the mirror. Then we can change the design of the mirror to improve this value of output impedance. And the last one in DC analysis, the D graphic, shows the load capability. We have here the output current of both drivers, and in the x-axis we have the sweep in the load resistor. Then the result is a deviation of output current for Howland in around 3 kilo ohms of load. But for CMOS, we cannot verify any deviation until 10 kilo ohms, which is, shows a high load capability for this kind of structure. We done a comparison using AC analysis for the output impedance of those drivers. And you can verify that for the frequency of interest of our study, and beginning in 100 kilohertz and 1 megahertz, we have a difference in output impedance of 1 decade, approximately. So, this difference influences. There's there is a low influence of this difference in transient analysis, for example, because we have a pulse of trains and this is a CC in that interval of time. But this difference will make difference in the transitions of the pulses. If the transition is fast, in spectral analysis, the difference of output impedance will appear out of the band of interest, the bandwidth of the interest. In spectral analysis, we can verify for a nominal condition for that load we presented that both circuits are equivalent in analysis. They are all equivalent in the generating the output current, the measured voltage, and for impedance measurement in ABS impedance or in phase. So we can conclude that the switched CMOS current source could be a better choice for time domain spectral analysis, time bioimpedance spectroscopy for the VLSI design or integrated circuit design. Some advantage from the Janssen current source is low power consumption, low supply voltage, high output dynamic range, high output impedance that is programmable by design, high output load capability, and output current programmable and independent of the input signal. In addition, if we can design a precise current source, we can eliminate the current measurement circuits, which make the circuits for wearables low power consumption, diminish the power consumption of the measurement part of the front end. It permits tetrapolar measurements, and for future works we have a lack of design equations for this kind of structure to make optimize this design of this current driver. So thank you for UDESC, FAPESC, CAPES, and you for your attention. Thank you.
We have 1 hour for question. Just kidding. No 1 hour. Just few questions.
Thank you for a very interesting presentation. I saw that your circuit was AC coupled and had an AC coupled output, and you have a very narrow range of frequency, a very narrow bandwidth from 100 kilohertz to 1 megahertz. What is the reason for this narrow bandwidth, and can the circuit be used outside that bandwidth?
The narrow bandwidth or the chosen bandwidth and the chosen electrode schemes distribution was inspired in project of our group. So we use that bandwidth and we use 4 bandwidths of 1 decade of interval. And for a complete spectrum, we usually do 4 analyses, and after we compare these results to make the whole spectrum. Actually, we can make 4 starts in 1 kilohertz and terminates in 1 megahertz or even so.
Okay, thank you.
Any further question?
Hello. When did I'm not an engineer, number one, and so when did the Howland, if I'm saying that correct, did it outperform the other one in any instances?
I don't know if I understand your question. If the Howland is if the switched CMOS is better in every aspect compared with the Howland source.
Yes.
Okay. The Howland source, I think, could be more suitable for general usage. What I mean is, I can design and implement it with discrete electronics, but the SCMOS is not suitable for this. It's optimized for integrated circuit design.
I see.
The Howland current source could be used for both the frequency domain analysis or for time domain analysis. The switched CMOS is optimized by time domain analysis using binary pseudo noise signals.
I see. Okay. And so, when choosing one or the other, wouldn't it also have to do with the application that you're focusing on? And I think you kind of answered that, but maybe you could expand on it a little bit.
About the applications.
Yes.
The Howland current source, I think, is largely used due to its generality. So it could be designed for almost every application in impedance spectroscopy. But we have some remarks to verify in this application. For example, for fast transition signals, fast post transitions, the op amps will become very expensive because they should be special application op amps. For low voltage design, those in market, I mean in the market of op amps, when we can buy an op amp, will be difficult to match velocity and low power supply. And for example, wearables or battery supplied circuits, the op amps, the tendency is to meet in the market slow op amps for the transition or very high frequency excitation signals. This is some trademarks you should verify to use the Howland current source for high-speed signals.
Any further question? Just give some hints. We have been using DIBs, digital interleaved binary sequencing. It looks like a core bar of a product that you buy in a supermarket. This is designed to be a electrical impedance spectroscopy based on time, not on frequency. So that means quicker. And we have a big research in German called Wbio, who works a lot with time domain. So if you want to you want to pick up a tiny amount of signal in a very quick way, like genes, like DNA, like some, you have to use time domain. And for time domain, it's better to use DIBs. DIBs is good, but not for commercial unpops. You need velocity. So you need special ones, and special, that means money, but also you are looking for consumption of energy, and you don't have both of them. Wide bandwidth, lower power, no possible. So using this structure, we are going to achieve wider bandwidth and small power and time domain. That's three things. Okay, thank you. Any further question? No? So thank you very much, Pablo. And the next one is not coming. Thank you.
Thank you.
And we are free for lunch. I am going to set you free, okay? So if one of you would like to visit my lab, you are very welcome to join me. Yeah, we are in a building, but the campus has a lot of buildings here. One of them is the Department of Electrical Engineering, where you can find a lot of labs, but including my one as well. So not my lab, the one I coordinate, okay? I don't have nothing here, just a pen. This pen is mine. Thank you. Oh, if you want to go to the lunch in the shopping center, you just need to go down to the building, turn right, go up to the end, turn right again, go to the hill, left shopping center. The gate is exclusively open for us. Not for this event, to the university, okay? It closes at 10 o'clock, by the way, p.m. Vai fechar?
Não, fica aberto.
Vai ficar aberto? No. It's going to be open. So it's better you keep it. Okay. If it's heavy, I can help you. Okay, you said it. Have you brought some drinks? Yes. Dr. Pedro, the Sherpa. program of the event. The next lecture will be given by Dr. José Pedro Serbena from the Federal Technical University of Paraná. Take the stage.
Alô? Tá bom? Tá bom? Sim. Tá. Good afternoon. Actually, it's Universidade Federal do Paraná, not technological one. We have this one too. There are two federal institutions, the technological and the non-technological. So I'm from the non-technological, but that's okay. My name is José Serbena. I'm from the physics department in the Universidade Federal do Paraná in Curitiba, Brazil. I will talk about volatile organic compound detection with nanostructure embedded resistive sensors, which is a research line of our group, actually. This is just the outline. I will spend one slide just showing on what we work in our research group. Then I will move on to the motivation of this work, the experimental procedures, and some examples of sensors we fabricated and characterized, and some conclusions. Okay. Well, our group is the group of organic optoelectronic devices. I think that the letters here are too small for you to read, but it's in English it's called good. It's a joke. Actually, the group is dates from 1994 from former Professor Ivo, and now I am the one who is coordinating this group. So we started working with organic semiconductors and applying these organic semiconductors in optoelectronic devices. So we have some research areas that we recently are less studying them, but they involve charge transport and injection in organic devices and materials, synthesis of conjugated polymers and nanostructures. So we are not from chemistry, but we do only the simplest synthesis. We have some works in organic thermoelectrics devices and materials. But our main research lines relies on organic field effect transistors. So we are working both mainly with the electrolyte gated transistors and the organic electrochemical transistors. In the geometry, we do the usual planar architecture, and also the vertical one, which is less common, but it can be done very well with organic materials. And there's a research line on VOC sensors, which are the volatile organic compound sensors. So these sensors are gas sensors, right? So basically, in this research line, we try to figure out materials and nanostructures that can have the potential to be used as sensing material or active material in the sensors. So we do very few things with thin films, and we work more on these nanostructure devices. And that's why the kind of things that I will show you in this talk. Okay. Actually, the organic electrochemical transistors and the electrolyte gated field effect transistors can also be worked as sensors. And there's a part of our group that is trying to join this these three research lines in one. So that's a step that we are giving. These electrolyte gated transistors can be more useful for liquid analytes or sensing liquid medium, and these nanostructured sensors for gas medium. Well, the motivation of our work relies mainly on the materials that can be used for the sensing, for doing sensors. We know that as the day passes, more and more sensors are coming to our lives in wearable devices, smartwatches, cell phones, Internet of Things. So usually these kind of sensors, they rely on thin films and inorganic semiconductors and organic electronics. So our main motivation is to try to use nanostructured materials and nanomaterials incorporate in this kind of device. So our focus beyond that on the material science is to try to develop room temperature gas sensors, because if you look at the transition metal oxide sensors based on thin films, they need to be heated above 100 and 150 degrees Celsius. So you have to put some extra energy just to activate the sensor. And we try to investigate materials that can be used for room temperature sensing without this necessary energy this extra energy for the device to work. So the volatile organic compounds are organic chemical that evaporates at normal indoor atmospheric conditions of temperature and pressure, which means that if we have one of these substances in liquid form and we just leave the bottle open, it will evaporate. So it will diffuse to the atmospheric ambient. And this can be a problem if this compound is toxic for humans, for example. So if you have some kind of industry that use formaldehyde, for example, you have a wood industry, you can have the contamination of the ambient by this kind of toxic compound. So it is important to monitor and real-time monitoring of these toxic substances to prevent some kind of injury on the workers. So our analytes, or what we try to sense, are these kind of substances that evaporate very fast or very easily. So because there's an easy of handling these substances and easy of measuring them also. Okay, so actually we do not work with biological things or biosensing, but our main focus would be in this kind of substances that can harm the living things. Okay. Well, just really fast. This slide is some kind of primitive sensors for methane gas detection in coal mines. The first detectors were with human leads. People use it to detect that methane because if it gave some kind of explosion, that means that the methane was there. But it was not a good detector because the holder probably would get injured also. Then another sensor that was developed was the canary bird. So the canary bird would inhale less amount of toxic substance. So with less substance than the human, he will die. So if the canary bird would die, then people know they should leave immediately the coal mine. This is also a detector, primitive detector. And another one is the flame light. And this one would, the flame, the color and the shape of the flame would depend on the substance that was in the coal mine. So, for example, if there's too much, there's no oxygen or oxygen is getting short, then the miners would know because the flame would change. So they knew they would need to leave. So these are three kinds of primitive gas sensors that were developed by humans. Nowadays, we have the development of electronics and the integrated circuit, and this allowed many modern applications and many other developments of sensors. We have sensors for sport equipment monitoring, like smartwatches, and the health monitor, which are more clinical, different than these that we wear. We have sensors for food quality, smart agriculture. So Brazil is strong in the agro business is really strong in Brazil, and it is strong because it uses a lot of sensors and a lot of technologies. Air quality, mining, oncology, and measuring CO2 on Earth. So there are many applications for sensors in measuring gases. Okay, so there's an important part to be developed in terms of technologies. And within this, of course we have some commercial devices to make this gas sensing, but we also need to work on the materials, the active materials that will do the sensing, which will transduce the signal. Okay, and that is our focus. The focus of our group is to focus on these materials. Okay, related to the biosensing, we can have some applications also of gas sensing on the human health, for example. So there are some works that show that patients with different kind of disease, they exhale different gases, specifically some organic volatile compounds, and this kind of measurement, if it is selective, if it has enough selectivity, you can use it to monitor if the patient is ill or not, and maybe diagnose some kind of illness. So it's not a commercial, it's not an easy thing to do, but it can be done, this kind of biosensing with gas sensors also. Okay, but that's not what I will show. Okay, and finally, our goals, our research are directed with the Sustainable Development Goals from the United Nations, mainly related to good health and well-being. In the case of sensing compounds that are toxic to humans, and for responsible consumption and production, because we use very few amounts of material to do our sensors, and we also have some kind of projects that work on reused materials for the active as active material. Okay. Well, so basically, the gas sensor, the chemi-resistive sensors, how they work. You have some kind of gas molecules. It can it can have some different molecules here. One of them will be the analyte, and you have the sensing material that will interact with this gas molecule. Okay, this interaction will lead to changes in some physical quantity, like for example temperature or conductivity or etc. And we can measure these changes using some electrical signal. In here it is represented capacitance, inductance, and resistance. So the idea is that we have a relation between the interaction of the gas molecule with the sensing material and the electronic signal we measured. So we just measure the electronic signal here under an controlled ambient and controlled environment. So we choose the molecules, or the best we can do is they choose the analytes and the molecules that will interact with the sensor device. And so this interaction usually depends on the absorbed molecules on the surface of this sensing material. And in addition to have this electrical signal to be collected, we need to have a percolation paths of this. In this case, these are nanoparticles. They need to touch each other, and they need to touch the electrodes of the substrate here to measure the electric signal. And this is an example of the structure we use in our sensors. They are just interdigitated electrodes. These are gold on top of the fiberglass FR4. And we have this the sensing material, which in our case are nanostructures. I would not call thin films of nanostructures because they are not. They are just randomly deposited nanostructures in our case. And we have the chemical gas to do the interaction. Okay, so what we measure is the impedance, is the electric signal along this pair of electrodes, and we try to correlate this electric signal with the gas. So in a general way, what we do is to use different nanostructures on sensors. So we do not, at least in this presentation, I will not show you the synthesis of these nanostructures because they were synthesized by collaborators. So when we synthesize the nanostructure, we need to do some materials characterization. So I will skip that also, and I will move on directly to the sensor fabrication. Okay. In all cases, what we do is to take the nanostructures and disperse the nanostructures, and it's not a solvent; it's a liquid. After that, we fabricate the sensor, we deposit on the substrate, and we do the measurement, and finally we analyze the results of our measurements. Okay, so this is the basic procedure that we do, and in the case of all nanostructures, we do this same kind of methodology. Okay, so it will be repeated. And for the dispersions, we use this. This doesn't matter the amount of nanostructures, but the important thing is that we put a surfactant to avoid the bigger structures. So we have this procedure to separate the bigger structures from the really nanostructures. So we just use the supernatant part of this dispersion, in which lies the smaller particles. The bigger particles, after these 2 days and after the interaction with the surfactant, they will decantate to the bottom of the flasks. So we are using only the smallest ones. Okay. And prior to the deposition of that dispersion, we have this cleaning procedure, and this is just an image of our substrate. And what we do is to drop a droplet of that dispersion in this substrate, wait it dry, the solvent dry, and hope that a path for the current, electric current, or for the electric signal appears here between the pair of electrodes. Okay, so this is not a well controlled deposition process, and that's why we do not use the signal, the absolute value of the signal. We also use the relative change in the signal in our samples, because since we do not control the morphology of this dispersion after it dries, the absolute value of the signal will be different every time, but the relative change will be more similar. Okay, so this is a drawing. It's drawn like a thin film, but it's not a thin film. Okay. And the measurement we do we put these sensors in a sealed vessel, and sometimes these sealed vessels goes into a glove box to have a better control of the ambient, and we measure the impedance through an LCR meter. Actually, we use it in these works only the real part of the impedance. So this R means real part of the impedance, and we thought it would be enough to show the sensing of these devices, that they work using only the real part of impedance. And now in the last year we are using also the imaginary part and doing more with this data. Okay, but we can do a lot of things with just the real part of this impedance. This is another drawing and another picture of our system. So here's the vessel, and it is inside a glove box, and our inert gas is nitrogen. It's not dried nitrogen. It has some filters here to control the humidity, but it's not like a low humidity environment. It's just a controlled one with like 50% or 15% relative humidity. Okay, in this chamber. Okay. And this is the methodology we use to characterize the sensors. So we measure firstly the spectrum of the impedance, and we use only the real part of it. After that, we try to figure out the signal to noise ratio and the sensitivity of the sensor. And this parameter is the main parameter we use to compare with other devices in literature and devices of ourselves in the group. And we also determine the limit of detection and response and recovery times, and the selectivity we test changing different analytes here. So it's not a really fine, good way to test selectivity, but it at least is an initial approach. Okay. So the first material I would like to show you was provided for us by the Center of Excellence in Strong Materials. Is a collaboration with Professor Neil Colville in South Africa, University of Witwatersrand, and they send us some hollow carbon spheres and some hollow carbon spheres doped with nitrogen. So in this picture here is just a transmission electron microscopy of these hollow carbon spheres, and they have almost 500 nanometers in size. And the difference is that during the synthesis they can dope with nitrogen, less amount, and a more amount of nitrogen. And the idea is that is to see if we can tune sensitivity with this kind of dopants, because we will introduce more defects in the structure, and these defects will be the active sites during the interaction of the gas with the nanomaterial. So what we did with these nanostructures, we did this dispersions and the thin film, the position of them, and we measured the impedance as a function of frequency for different concentrations of different analytes. In this case, ammonia is the analyte, and we can see that there are some changes in the impedance of this, the real value of impedance, with the frequency and also with the concentration of the analyte. So what is the advantage to do this kind of measurement is that we can choose the frequency that best works for the analyte and the nanostructure. So basically this spectrum here is for us to choose in which frequency it will be done the rest of the analysis at this point. Okay, and in all the cases we can see that in here is not so evident, but there are changes in the real value of impedance, and for this guy here doped with less amount of nitrogen, there is a more change in the resistance. But in addition, the value of resistance is lower also. So that's why we don't use the absolute value of resistance. We do relative changes here. I will show in the next slide. And to choose the frequency, we also do the signal to noise ratio. For this, some of these frequencies, we measure the blank, or the signal, the electric signal in the inert atmosphere. This is the signal of the blank in the for this kind of measurement, and we do a ratio of the signal measured with this signal of the blank. So we use these two parameters to choose the frequency, the optimum frequency of operation, because as we see, if we go up in frequency, this signal to noise ratio becomes higher, but then the signal is less dependent on the analyte concentration. So we have to choose between these two competitive frequencies. Okay, if we go to lower frequencies, we have less signal to noise ratio, but improving difference here. Okay, and we also, with the signal of the blank, we determine the limit of detection of each sensor. We use the parameter of 3 sigma, 3 times the deviation of the blank, and with this we have a good estimative of the limit of detection of this of our sensors. So, and this is another parameter we use to compare with devices in literature. Okay, and finally, the sensitive. The sensitive, we do not use the absolute value of the impedance. Because mainly our way to deposit the materials is not so homogeneous. So every time we have a sensor, it can have different resistance, and we need to see how it changes relatively to the resistance with the inert gas, with the blank gas. So to measure the sensitivity, we use this delta R over R0, where R0 is the impedance on the inert gas, and delta R is the difference between the impedance on the specific analyte concentration and the inert gas. And we do another approximation, which is consider that these guys are linear, which is not always the case. So, but this is a fast way to compare the device and see at least if it has the potential to be used as active material for a kind of analyte. And the sensitivity is essentially the angular coefficient of these curves. Okay. This is for ammonia. Okay, I think it's. I wanted to show the water also, but I think it. I have missed the slides. I don't know why. Doesn't matter. We also test with water. This the same procedure. Okay, because water sometimes plays an important role in the sensing mechanism of these nanomaterials. And what we see that for this kind of hollow carbon spheres, they are more sensitive to water than to ammonia, for example. And also in addition, but the more the hollow carbon spheres more doped with nitrogen has a sensitivity higher for ammonia. Sorry, this is LOD. This is not higher, but the water here. No, it's sorry, my mistake. I think it's okay. It's black. The LOD, the smaller, the better. Okay. So yeah, in this case, the water is it's not good for the sensing for the functioning of this kind of device. if we want to apply this sensor on a real environment, we need to take care of water, because in a rainy day, certainly the humidity will be much higher than in a day that is not raining. So that's why we test in water. And we see that water is no good. It interacts a lot with these hollow carbon spheres. Okay, and here is just the parameters, a table with parameters in which we detach, or we highlight the best device that we were able to produce. In this case, for ammonia detection and for water detection, the non-doped hollow carbon spheres are the better. Okay, so sometimes the doping process doesn't have what we expected, and sometimes it does. But we test on other analytes also, and by this way we can try to find some analyte that is that has a better sensitivity for the specific material. We usually test with methanol, chloroform, toluene, acetone, water, ammonia, and lactic acid, and we see that this there's a big variation of these analytes with these solvent molecules with the nanostructure. And in this radar graph, it is the limit of detection. So we also measure for this the same analytes. Okay. The second nanostructure I wanted to show you is the gallium nitride nanostructures. In this case, they are smaller. They have around 19 nanometers, and this gallium nitride is a semiconductor, and it was already used in gas sensing, but mainly in the form of thin films. So in our case, we are using the nanostructure form, and in this case they were not doped, but they were coated with carbon. So during the process of synthesis is if I'm not mistaken, is a CVD process. You can put some carrier gas, and if the carrier gas is argon, you have higher coating of carbon on the surface of the gallium nitride, and if you use nitrogen as a carrier gas, you can have a thinner coating, a thinner surface here, a thinner layer of carbon on the surface of this gallium nitride nanostructure. And the same, the idea here is to, in this case, if this coating process can improve the bare material here, okay? Since the gas sensing is a surface phenomenon, okay? And we did the same procedure. We firstly do the spectrum, the impedance spectrum, and we, in this case, are analyzing just the real part of the impedance, and we measure the signal to noise ratio and choose the operating frequency or optimal operating frequency for each of these devices. And we also determine the limit of detection and the sensitivity using the relative change in impedance. Okay, And in this case, we also did with ammonia and water, and usually in the previous material, water had more sensitivity than ammonia, and in this case, water has less sensitivity than ammonia, which is good for if we want to try to test this sensor in uncontrolled ambient or uncontrolled atmosphere. And what we see is that in addition the coating with carbon improves the sensitivity of this of the device. And here is the limit of detection for ammonia and for water. And we also did the test with other analytes, and if we want to detect a specific analyte, of course there's in this case we cannot detect mixtures, right, because they have this cross selectivity, this they have the signal for many analytes. So to investigate selectivity, it would be another challenge, okay, another focus. And finally, the response and recovery times. We choose the operating frequency, the optimal operating frequency, and we do a time measurement. Leave that in the we put the device to be measured in time, and we expose the sensor to the ambient with the analyte, and when we open here, it's inside the glove box, it's in the inert atmosphere. So we put the gas on, or the analyte on and out. So what we measure is the real part of impedance, and we also can have the signal here in time. And in this case, we also measure the response time, because depending on the application, it will be important to know if it is a fast device or a slower device. If the gas is too much toxic, then we need, and we have real-time application, real-time monitoring, and we need a fast response sensor. But if it's not, if we are in, for example, in a building of an industry with some ventilation, and we don't need to have this fast response. If we have a response of minutes, at least we can detect some kind of leakage or something like that. So this is important for us to try to figure out for which application this material, this sensor would be better. So we measure the response time at the 90% of this saturation value, and the recovery time here at 10% of this saturation value here in the blank. And usually our sensors, they are not too fast. They rely here in the seconds, hundreds of seconds. So it is around 1 minute, 2 minutes to get a signal, a saturation signal. So at least we know for that if we need something really fast, we cannot use with these sensors, but it can, if we done in a cheap way, it can be installed on a big building, several of these, and monitor real-time monitoring an ambient with not deadly gas, for example. Okay, And what we see is that the doping and the coating, they change the interaction of these analyte molecules with this active material. Okay. And finally, the last nanostructure is this study was made in collaboration with Professor Christine in Université de Lyon. She did some hexagonal boron nitride nanostructures or nanoflakes, and she doped it with this barium fluoride salt. This material, this HBN, is used a lot in electronics. It's a high band gap semiconductor. It's really stable, and usually it's used as substrate to study graphene. So we didn't thought it would serve as a sensing material because of its properties. But if we introduce some defects in this structure, usually by doping it, we can have some sensing. We can turn on this material into an active material. So the idea was to compare these flakes with the bar HBN and one doped with this BAF2. So we have 4 contents here, and this is just a transmission electron microscopy image to show its size, which is much bigger than the previous one. And we tested these materials in the sensors, and we did for different analytes also. And here's the measurements of the real part of impedance, and this is the characterization of the table showing the results for acetone and ethanol. And what we see is that for less amount of material of this doping process, it has a higher sensitivity, and if we increase, what happens is that we change the structure of the hexagonal boron nitride, and the structure becomes more compact. So what we are doing, increasing this doping content, is to changing the way or difficulting the way that molecules of the gas can reach the active sites of the HBN. So actually, the bare one is better than the doped one, but the small doping amount here was the best. And just to compare with other devices in the literature, this is concentration range of this work, and it is room temperature. And many others, they usually need temperature to become active. And this is the sensing mechanism we proposed. Usually it starts with the absorbed oxygen in the surface of the material. So there is some charge exchange, and we have these electrons in the surface, and depletion of electrons in the material. So this kind of thing will enhance this sensitivity of the analytes, and what happens is that the analyte interacts with this absorbed oxygens, and usually the process ends on the formation of CO2 or CO and water. So this is the sensing mechanism we proposed for this device. And to end, we also tested these sensors after 18 months and realized that the 18 months start in ambient conditions. So we realized that they were quite stable, because our experience is that usually materials degrade after 2 or 3 days in the ambient, start in the ambient conditions. And this one, after 18 months, we were still able to get some signal here and make the measurements. So in terms of stability, this kind of material, it would need less research effort to produce a stable material. We're using this one. Okay, so in conclusion, to be brief, we were able to use or show that different carbon nanostructures, doped, and gallium nitride nanostructures coated with carbon can be used as active material in the gas sensors. And we also showed that this hexagonal boron nitride also can be used as active material in gas sensing, and it is really stable. Okay. So I would like to thank you for the patience to watch it full, the funding agencies, and for those who have time, our lab is here in Curitiba, which is around 2 hours trip from Joinville. So we can go by car or by bus. There are some nice places to visit, and we are off the coast, so if you want to go to the beach, it's better to stay here in Santa Catarina. This is the university in our campus. It's the first university founded in Brazil. And thank you for your attention. And this is Clara, who did the most of this work. Okay, thank you.
So, thank you, Serbena. And we have a couple of minutes for questions.
Yeah, thank you for a very interesting presentation. It's more a comment than a question. You said that you had mainly only measured the resistance and not the reactance, the real part of the impedance you have measured. But you were thinking about measuring the imaginary part. But according to the theory of Kramers and Kronig, there is no extra information in the imaginary part. So if you have good measurements of the resistance as a function of frequency, you have all the information actually that there is, and there is no extra information in the reactance.
What we want to do is to use the real part and imaginary part of the impedance to build equivalent circuits and try to relate them with what is happening in our systems. So this, when I was talking about, additional information, I was thinking on this, because sometimes we use polymers with these nanostructures, and we use carbon nanotubes with polymers with nanostructures. So the idea is that with analyzing the whole signal of impedance spectroscopy, I mean, I cannot do this only with the real part. Do a equivalent circuit model, right? At least I don't know how. I just
Yeah, you can.
I-
Actually, it's possible because you can, from the how the resistance varies with the frequency, you can calculate the imaginary part. So you can, I mean, if the resistance is flat, you know it's purely resistive. If it goes down one decade by decade, then it's purely capacitive. And if it's between, so it's possible. It's possible from the resistance to calculate the reactance.
okay, then
Yeah.
I will talk to you in the coffee break.
We can, yeah.
Any other question? Okay.
Hello, thank you. And so to use the platform, can you reuse it? Can you wash it? Like how would you do a second test?
What we do is to, when we just start testing the nanostructure, we wash the substrate and we just see if it's okay. If the material goes out, and we do the tests with this washed substrate. But when we are done with dealing with the parameters, then we do tests with fresh substrates. Actually, they are cheap. We pay like R$2 for each of these substrates, so they are not expensive. I think that the nanomaterials are more expensive than the substrate. But sometimes we can use, sometimes not. Depend on the nanostructure. We need to test because sometimes they do not leave the substrate. Okay. If we use, for example, titanium dioxide nanoparticles, they are removed very easily. Zinc oxide also.
Thank you.
I have a question. Anyone can answer. If you are measuring imaginary part of the impedance, it's not the right way of doing if you are looking at the dispersion of the electrical field rather than measuring only the resistive part of it? Don't people use phase angle for food quality control once you have the imaginary part of it? Because according to my equation, according to the laws, Maxwell, arctangent of imaginary part divided by the real part of it. So don't you really need to measure the imaginary part? Just a question, and I'm open for anyone.
As I see, we have the three parameters when measuring impedance. We have the real part, imaginary part, and frequency. So I think we have more information and more degrees of freedom to deal with the data. As I see, I think it's better than measuring DC form, for example, when you just apply voltage and measure current, so you have the resistance or resistivity. But so in DC you don't have frequency, neither these two parameters. So that's my that's my I don't know, my feeling on this.
I have a question. You changed the structure, the atomic structure of the crystals there. So, considering that, you inserted defects and but it continues causal. There is causality because it's a natural material and synthesized, but it comes from nature. So causality is still maintained. The problem, I believe, in calculating would be the algorithms that may not converge easily, depending on the impedance. But in this case, only with resistance you can interpret the results. But what I saw while doing that, trying to avoid obtaining resistance instead of reactance, would be the choice of the right algorithm to converge to the correct value. Well, but you can do the analytical expression of the Kramers-Kronig relations, but that would be more an effort. I believe that somehow we can experimentally deviate from that. That's an impression.
Yes, I totally agree with that, and it's often easier to measure it than to calculate it. But I just wanted to make the point that it has been shown by Kramers and Kronig, and it's used in optics and it's used in electrical engineering, that there is a relationship between the real part and the frequency response of the real part and the frequency response of the imaginary part. So you can, from the impedance modulus, you can calculate the phase angle, or you can calculate the imaginary part from the real part, if you know the behavior of the real part over a very wide frequency range, and that's the limitation. You have to know the frequency response outside the frequency range that you want to calculate in. So there are some weaknesses. So it's often simpler to measure it. That's true. But you can use it as a test if your measurements are good also. Because the imaginary part mainly looks like the derivative of the real part. If the real part is flat, the imaginary part is zero. If the real part goes down, then the imaginary part is negative, and so on. So it looks like you take the real part and you derivate it, or you differentiate it one time, and then you have the curve of the imaginary part. So, okay.
Okay.
Any further comment, question? No? Thank you very much. And the next talk will be given by Professor Alexander Sadi Paterno, and it will be given in a form of a tutorial.
Good afternoon. So I am going to talk about, that is a work, not a work exactly, but a presentation on topics that may well integrate part of what has been said here. We can join everything that has been said, and some aspects of those ideas will appear somehow in this application. So, the first motivation for this work would be what has been said in the beginning by André. He told about the project that we would try to develop a platform to allow those antibodies that he would participate in the design, so that we could detect those antibodies using the infrastructure of this platform. Well, the first step towards this development was after a discussion with the group where the biomolecular people were there, and they told that one first step would be to develop a surface plasmon resonance detection of those biomolecules. Well, a platform was an ongoing development in the literature that people are using a very small platform, which is the optical fiber, covered with the materials where we excite the surface plasmons, which would be a thin film or nanoparticles, for example, metallic nanoparticles. Well, this is the motivation of the design of the platform. Let me see, where is the pointer here? So, and you are going to see in some ideas that are going to be presented, and this will also allow the students here in the university to take part in some of the works where we are going to show how we fabricate those devices and actually platforms that we are going to present. And you are going to see also that material is also some critical aspect. When we work with optical fiber sensors or fiber optic sensors, we think immediately of using a standard single mode fiber, usually, because it's the most used fiber optics, which is the optical fiber that we use to transmit information all over the world, which is the most used optical fiber to do that. But in this case, it will be seen that the material will allow an improvement in sensitivity if you choose, for example, polymeric materials. In addition to that, we have the benefit of improving biocompatibility. So, biocompatibility is also some aspect that the student or the interested researcher must worry about. Well, the first question then will be, is such platform a biocompatible platform? Well, we are not going to use in contact with tissue or living organisms. We are going to use as a point of care, as it was a first expectation of this work. Well, we are going to use it as a label-free detection system that is expected to be developed as a label-free detection sensor, and it must be somehow then it may lose these characteristics of label-free detection. It may be functionalized, and so that we can, while using some optical or electrical means, we can or try to indicate the presence of some biomolecule, specific biomolecule. And of course, there is another aspect, which is the interrogation of the optical fibers, and also how to read the electrical, electronic, and optical sensor, which is a work that is not going to be discussed here. We are not going to talk about interrogation, which would take more time to discuss that. Well, as you are probably aware of, we have to worry about safety and accuracy, and also repeatability of using such devices. But this is a specification that may or may not be attained. So, and also after eventually we can think of using such sensors as an in vivo application. So that is an ambition, but we may have the luck to direct our efforts to that. Well, I'm going to, for those who are not aware of, but for those who took the course on waveguide theory, I'm only indicating here basic principles qualitatively of how the specific waveguide that we are going to use, well, the sensor is a waveguide, and the material of the sensor is a dielectric material. So it has no conductivity, so non-conductive material. It's silica mainly. As a first step, we tested in a previous experiment done by one of the students here, Afonso. We developed a sensor on an optical fiber, a silica optical fiber, ordinary multi-mode silica optical fiber. What is a multi-mode? If you remember that this is a cylindrical tube here with a core, with a material, the electric material, that has a refractive index a little bit higher than the cladding, which is the material surrounding the core. So the core is where the wave is going to propagate. So we are going to launch a wave inside this sensor by, for example, a laser or a broadband source, a LED. And this LED, depending on the size of this diameter in the core, you would have the possibility of have several angles of input into this core. So depending on this angle, and considering that we are going to modify a section of the fiber, this is part of the principle of what we are going to do to prepare the platform. We're going to change one section of the optical fiber. So you buy the optical fiber, a commercial optical fiber, or you buy some specifically designed optical fiber made out of plastic polymers, for example, and you remove one part of this cladding which surrounds the core. But in this case, it is illustrated here another method of reducing the cladding. We stretch the fiber while heating, or using a flame brush, for example. Stretching the fiber is going to reduce the size of the core. So the core is going to, depending on the reduction, it's going to disappear. And the new section of the fiber will have a different behavior in terms of propagation. It will be, as we call, a multi-mode section of the fiber. So there will be the possibility of several ways of propagating the wave in here. Why is that? It's because if we want to operate with surface plasmon resonance, we need some control of the angle of incident on the material that is going to be deposited here. Otherwise, we are not going to be very effective to excite the surface plasmon. So this is the idea. The core idea of this platform is the reduction of the fiber so that we can access the wave. So in this region, the wave that enters or hits the interface here will propagate a little bit above the surface, which with a wave, with a part of the wave that is called evanescent wave. So otherwise, if there is no possibility of accessing the evanescent wave, we will not have the possibility of exciting the surface plasmon. What is a surface plasmon? You're talking a lot about that, but we're going to see a little bit about that, not much mathematics to do, or physics, only a qualitative explanation on that. Well, the principle of this platform will be then a removal of the part of the optical fiber, either by etching with acid, either by reducing the size, or by polishing. We can polish also. In case of plastic or polymer optical fiber, we can polish and remove until we reach part of the core. So, what is a evanescent wave? This is an illustration took by from a book. Well, this is an illustration from a very old book which illustrates a coupler. Here you see the intensity of light propagating along an optical fiber. From here to here you have a section of the optical fiber. And in this region you have a reduction of the, actually you have a reduction of the diameter of the fiber, as we talked previously, along this section, and you have another fiber here. And this illustration here of the intensity of wave propagating on inside the other fiber is caused by a coupling. The wave is evanescent here, and this wave that is evanescent here is caused to couple to another fiber that is touching, they are together, stretched together. So this is a fused coupler. So the combination of two fibers here indicating that there is a coupling. So to indicate that there is the possibility of accessing the wave that is inside another fiber by only touching the other fiber, if it is prepared to allow the propagation of an evanescent wave. So in this case, of course, there is a rule here so that we know where there is the coupling, so that we can, if we cut here, we have no coupling, but if we cut here, it depends on the wavelength that you are going to use. But this is we call that as a couple mode theory. We are not going to talk about here. It's only to show that there is the possibility of accessing the evanescent wave. And we are going to do that. Imagine that instead of a second fiber, we deposit here a thin film or a layer of metallic nanoparticles. There will be the possibility of exciting surface plasmons. This is the first step. This is the illustration of the fiber. You use two fibers, ordinary fibers. This is a single mode fiber, and while cutting this region that is fused, you see the combination of two cores here. And well, how is it done? We first pick a pristine fiber or an ordinary fiber. We buy a fiber and we prepare that, we clean that, and we put them together somehow. There is a way of doing that. And we set it on a taper rig. This is a machine that we have here. Two motors or only one motor. This part of the fiber is fixed here by a fiber holder, and the second one also. And we have some heat source here. It can be a flame brush, but a heat source by a very stable and controlled gas torch. Well, this flame brush moves to and fro here in a very controlled way, so that we can, while heating this section of the fiber, we stretch it as if it were a cheese that you eat, for example, raclette, you know, the cheese that we eat in Italy. For the same reason, of course, not the same controlled way, not the same temperature, but you stretch it and you reduce the size. But we can also design the profile of the reduction. In our machine, we program that so that we can control that. Well, we can do the heat source using a laser also, a CO2 laser, carbon dioxide laser, and it performs the same way. While doing that with a laser, we can fabricate other devices that can also be operated to measure surface plasmons, as for example, long period grating inside the fiber by point imprinting inside the fiber with the CO2 laser the device that we want to produce, which is a diffraction grating inside the fiber. Well, this is a picture of the 13 centimeter taper. And you can see with our technique we can control the profile. This is an exponential profile and this is a linear profile. It should be linear, but we used an optical microscope with a controlled basis and took pictures, 160 pictures. And this, of course, is scaled. Otherwise we would not be able to see details on this profile. And this is perfectly exponential. So this reduction is exponential because of the programming of the movement of the flame brush that caused the heating. Why is that necessary? Well, for optical purposes, for example, we can design some dispersion curves here in this region. For some nonlinear optics, you can use those devices if the diameter is very small. For other reasons, in terms of fiber sensors, we can, for example, if we are going to use that as a acousto-optic sensor to measure the presence of biological material or biomolecules, we can design different diameters, as in a staircase design. So every staircase could have, for example, some antibody, for example. And considering that, if the signal traverses that region with a specific diameter, with a specific antibody, we can then match that signal with the specific response of the sensor. And then we can multiplex the sensor with different antibodies. So the idea previously proposed is a start to imagine how to do that with a different set of antibodies. In terms of random distribution of antibodies is something to think then. I wouldn't say that it's not possible, but with random distribution the problem is a little bit different. If you have a random distribution of hundreds of types of antibodies, for example, I don't know the amount of different types of antibodies that may happen in some immobilization that you may provide on the fiber. Well, the idea is you have the taper, the thin film or the material that is going to be excited and functionalized is on the fiber. The second step will be the same geometry will have the antibodies. That's what is already previously thought. Well, to illustrate the process, this is part of our technique. I'm going to unfortunately. Volta. Isso. Botão direito tem? Unfortunately, the video is not. Well, it's a simulation of the process so that you can be very aware of how this is going to be produced. Again, to integrate what has been said, this process of evaporating gold on a taper has been performed in Sebena's laboratory in Curitiba. So we started with gold. This was our first prototype in the beginning of the year. In January we did that. But the taper in this case was fabricated by using a multi-mode fiber which has a larger core diameter, 105 micrometers in diameter, and we removed 10 micrometers of the coating so that we can access directly the core, and it's a silica fiber. And we evaporated a thin film of 35 nanometers and also in sections. There is a reason why we did that. We used sections of thin films, 4 sections of thin film separated, not having contact between each other. Well, why to do that? This is another example, but now an illustration this is from the literature, where they did the removal of the plastic fiber and deposited a thin film of gold and immobilized the antibodies here. So, and of course they tested that with a micropipe and a pump. Of course there is a pump to insert the material that is going to be sensed. And they evaluated the surface plasmon spectrum. Well, what is that? Well, we obtained also this such spectrum. We didn't do the functionalization, but this spectrum shows some specific characteristics, as for example a change in the environment refractive index with a very large range of index variation, 0.1 index variation. And while it changes in gold films, you see that there is a change in the valley, the absorption here, a peak of the sensor, and it shifts to the red side of the spectrum. In this case they used a different fiber. This is a polymer fiber, so it has inside it different very lower refractive index in the core. So because of that, the sensitivity improved 26 times. So you see now that it is important to choose correctly for your application, for what sensitivity you need, the material of the fiber. In this case, simply choosing a silica fiber, you will operate in the visible range. So you will need material, equipment, devices that will interrogate the sensor in this. In terms, if you will need gratings inside the fiber, that may be a problem while fabricating the device, since it's more complicated to fabricate gratings or other types of sensors in this region of the spectrum. So what is traditional is to operate in the 1.3 micrometer and also in 1.5 micrometer range of the spectrum. It would be easier and much more practical. But of course, this interrogation of this sensor may be an application or an adaptation of systems to operate in the visible or in the infrared. So how it works? What is this surface plasmon? It produces, of course, a peak of absorption in the spectrum. If you launch a broadband source, one section of the spectrum will be absorbed more intensely. So in this case, you see that the peak of absorption will be shifted towards larger wavelengths. So in this case, this is for the plastic optical fiber. Well, how it happens? If you consider that the wave inside the fiber has a propagation constant, when this propagation constant matches the propagation constant of the wave in this, or the surface plasmon wave, you will have then the conditions, the parametric here conditions, where you will find the position of the peak absorption. But what is this surface plasmon? If you consider that metallic material has free electrons in its lattice, you will see that it will oscillate with the wave that is propagating, respecting this condition. And this oscillation will cause the pronounced absorption in the spectrum. Well, this is a qualitative and very direct explanation of this surface plasmon behavior. So that's what we would like to implement. Well, considering now this is practical that we did in January, we etched here. The development has been performed here at the university and in Curitiba, the Federal University, and also at the Federal Technological University, where the etching and the optical characterization with ultra broadband sources had been performed. We show here only the visible, but there is more information on the infrared. Well, what we did here, the student had a project from the course of waveguide theory. He designed the fiber holders that would allow the implementation of bonding wire on the thin film and also the etching of the fiber. Of course, the bonding wire, not the bonding wire, but the touching wire, thin wire, around so that we can have access to the electrical aspect of the electrode that had been deposited. So of course, since we worked with hydrofluoric acid, we needed a plastic that would be resistant to the acid, and we used a special printer that could print polyethylene filaments, and considering that, we were successful. Of course, the screws also were special. They were an alloy with nickel and steel. The fiber holder, so that it could survive the experiment and be used in sensing experiments, it should be prepared this way. So, this is a picture of this is the first prototype, and it worked. The first experiment to fabricate it worked. So, and the result. This is a photo from the previous publication that we presented in Campinas in May. This is the final version. So don't take pictures because the patent office told me not to show it. All right. Well, after etching, thermal evaporation. This is the glove box that we used in the in Curitiba to evaporate gold. And this is the first demonstration of what we measured, which was performed in Curitiba. There's a pronounced absorption here at 740 nanometers, which is different from the expected 640 nanometers of wavelength in a gold thin film, while exciting surface plasmon resonance. We suspect that there was a mixture of silver and gold. It's a blend. It's not a pure gold thin film. But this is the first prototype. And it operates. It can be performed also with silver. Why not? And this is a normalized spectrum. You see that we can easily detect the shifting by interrogating, for example, with a tuned laser here at this slope here of the spectrum, absorption spectrum. You can detect the shifting of this, if it is not, of course, very large. Otherwise, we must design other kinds of interrogation. So, that was a presentation in a format of tutorial so that you can have an idea of or have ideas on how to collaborate, and since we have several students to take part in these developments. And I thank first of all here the student who did the core part of the development, and those groups, and the projects that are financing this ideas. So I believe that's what we have to present.
So do you have any questions?
Thank you, Alexander. I just didn't understand how can you measure antibody-antigen interaction there?
Well, we expect some kind of selectivity in the behavior of the immobilization of the material biomolecules that are immobilized on the surface. We expect that. We don't know if there will be.
So you put the gold on top of the fiber, and then you bind protein onto the gold film.
Yes.
And then you put a patient sample, for instance, and if something binds to the protein that's there, it changes.
What is expected? First, so that the interaction is larger, we don't use exactly the tip of the fiber. We use along the fiber, some section, a few centimeters, for example. And the material is immobilized on the surface of it, on the surface of the film. We expect to have some change in the refractive index, the first step, but we know already that it's not sufficiently selective. We must have additional information. Why would we use an electrode as also the surface of the film? Because we would like to measure, for example, the electrical impedance. Of course, not at the same time, because when we inject current on the thin film, you will distort the spectrum, because changes will occur. It will heat the film, because the thin film, it's not prepared to be an electrode. The current must be very small. It will be more complicated to operate at the same time, but the electrode is there, so it is expected that it will work, and it has continuity. We measured there in Curitiba that it is continuous. This is a first try. People have been using as an optical sensor, so they select the material that they inject on the sensor. So if you inject blood, for example, it must be tested. I reviewed several papers where people used in the benchtop only the selected material. That's not sufficient, because the real case will be a soup of material, several types of biological material. Then it will cause, maybe it will not cause. With the idea of using the electrode, we can then expect some selectivity. This is one idea. There are other structures in terms of optical design that we can implement so that we can detect or try to improve selectivity. I believe in terms of engineering the design of this device, the selectivity is the most challenging aspect of this sensor. Selectivity, as you said, on the biological side or biomolecular side, is the selectivity of the specific antibody that binds to some antigen, for example. I don't know if I answered everything or your question somehow. All right.
Hi, Rek. Can you come back to your results comparing the LPG in silica and polymer?
Tá indo no PowerPoint.
Slow, okay. The other one, that one. Yeah. Let's say I cannot read the small figures. The graph on the left, the inset, it's varying from 400 to 800 or something like that.
590.
Okay. And what is the parameter that is changing from one resonance to another one?
It is the outside refractive index.
So you put different solutions and that
From
one.
1.3 up to 1.4.
1.3, 1.4, okay. And it's the same experiment on the polymer fiber is done on the right,
Yes.
on the right hand side. Okay. And in both cases, the LPG were grown by
In
the
this
CO2
case,
laser.
There is no LPG.
There's no
It's
LPG, only surface plasmon resonance.
Okay.
Okay. And have you made any experiment with the LPG coupled to the metallic layer, like Barbara's work in the past?
We have results on that, but
Okay.
not with biological material.
Yeah, but you have with different
Yes,
index.
it's with Ricardo Canuti in Curitiba.
Okay, that's okay. And only to maybe complement the answer to the previous question, there was a paper was published about 12 years ago, I don't remember, by the group of the Catholic University in Rio, and they have used it in a grating and a surface plasma resonance to identify the, let's say, dengue, to bind with the dengue virus. It was on the first appearance of dengue in Brazil, not the last one, but the previous one. It was a very interesting paper, but it was with the Fiocruz, but it didn't arrive to a final product to the market. That's a problem in Brazil always.
Anyone else?
Thank you for this talk. I was wondering if you determined the sensitivity of your platform in terms of refractive index units.
The sensitivity is very low in terms of the gold, considering that we use gold and silica. It is not the best one in terms of sensitivity. You can have with plastic fibers much larger, with a specific. Actually, they are based on fluoride material, and this will have a refractive index that is approximately 10% of that of less than silica. And this will cause, because it depends on the inside refractive index in combination with the metallic thin film. So that is a situation. What we are suggesting here is that the same film would be possibly used as an electrode. That's the suggestion.
Great. So, when it comes to biological applications, maybe you will need some microfluidics control. Are you planning to integrate microfluidics?
Using microfluidics, we use ordinary microfluidics material, and maybe they will be based on adapted cannules, small cannules, 200 micrometers thick cannules. So we heat that and we curve it so that we can have access and insert, for example, a deviation to insert a pump tube, so that we can insert the material. Well, actually, I'm not going to do that here. I'm moving to another university for a for a period, and then I will probably do that there. And the infrastructure there would allow the fabrication of those variants of microfluidics.
Thank you.
All right.
No question anymore. So we are done. Thank you very much. I have some news, good news. Will be a microbus or minibus, as you wish. Nanobus, no nanobus. Okay, micro or mini. Minibus departing at 4:15. 4:15. Okay, departing from the main entrance here. The place you guys came here this morning. So going to the hotel, because the driver must be here at exactly start another job at 5, and is very big. Is busy. Traffic jam is kind of nightmare at 5 o'clock. So if you want to go, otherwise you can stay here for longer, talking, chatting, visiting, and take a Uber. Uber, not taxi. Uber is cheaper than taxi. Okay, and there will be a place that we are inviting people to go for drinks. This is a pizzeria. They call que massa. Massa is the way of pasta. Okay, massa, pasta, the same. It's a rodizio. That means the food comes to you, and you don't need to go to fetch it. We'll get you right in the table. Pizza, all sorts, all type, and pasta, all sorts, all type, and a lot of. It's very delicious. My wife said that's the best pizzeria in Joinville. I said no, I prefer Italian pizzeria, but the people say that's the good one. So we will meet there at 8 o'clock tomorrow, and the price they ask you as a group for 79 reais. Yeah, I know that for us Brazilians it's kind of a nightmare. Yeah, 79, but it's very good, very high quality pizza, and from 8 p.m. So if you want to join, we are going to put the direction in the website. I think it's kind of close to the hotel. No, kind of walk, 10 minutes walking, something like that. Okay. And another place is Mirante. Mirante is a nice place that you can be. Is a very there's a hill, big hill. You can walk there. I think you've been there once. Yeah, and there's a tower in spiral way, and you goes up, and you can see where Joinville is. You can see actually we are close to the sea. There's a mangrove, rivers, and it's very high. I'm afraid today is not good because we have cloudy. This is June, weather for you, but early in the morning for the sunrise is spectacular. It was supposed to be good today, sunset. People usually go to sunset and sunrise. That's a good place because you're going to arrive in the hotel so early and it will be so boring. But the place you are in that street in front of the hotel, there's a lot of restaurants. It's the busiest road in Joinville. A lot of restaurants, bars, danceteria. There's a big Mexican food there as well. You can have tacos, okay? Tacos there. Pizzeria, all sorts of type, and dance if you want to, and drink, okay? And now you are welcome to the coffee break. Thank you.